Okay, so in relation to pharmaceutical biotechnology, what will all of this information mean? What will be the impact of genomic sciences, not only on biology, but on the way we conduct drug discovery and drug design? Well, certainly knowing the composition of humans gives us insight into our individuality. I notice that each one of you is unique, but you are 99.9% the same at the DNA level. So even though I can tell each one of you is unique, most of us share way more in common than we do differences. But it's those differences that make us unique. Keep in mind, too, that skin color is one gene. I think this is a remarkable fact, seeing that much is made out of this characteristic globally, but the SCL24A5 gene is what codes for pale skin in Northern Europeans. Big deal, really. Owing to the fact that this makes them more sensitive to sunlight and therefore can probably make vitamin D better. That's about that. But what are the implications of knowing all the genes in humans? I said that landing on the moon pales in significance because knowing all the genes in humans and their relatives has vastly affected how we think about anthropology, where anthropology may have previously been mostly archaeologically related to recovered bones and artifacts, shards of clay, arrowheads, and cave paintings, certainly give us insight. But knowing DNA has allowed us to compare ourselves to other closely related species, and we find that, in fact, we have admixture from all over the place. Homo sapiens is apparently a combination of various other closely related organisms. We now know that Neanderthals probably did mate with Homo sapiens. We know this because we have the sequence of Neanderthal DNA now, and we can compare that to the sequence of Homo sapiens. We also know the relationship amongst humans and can compare that against migratory pathways that were previously assumed by artifacts. It certainly has impacted the way we do pharmacy, and we'll get more into that in a minute. How we conduct medicine at a personal level, but also aspects that are now coming of age, such as stem cell therapy, tissue engineering, gene therapy, xenotransplantation, all affected by DNA technologies. It has affected and will continue to affect the science of psychology, where Jung and Freud might have said it was mostly your environment, maybe something that your mother said to you that made you this way. We now understand that there is a deep impact of our biology on certain behavioral functions. A friend of mine even says that perhaps all psychology departments should be in the biology department from now on. Well, he's obviously a biologist, and I'm certain that many of my psychology friends would disagree. But it's also the way that genomics has affected pharmacy. We can now understand how it will also similarly affect psychiatry and the ability to treat disorders with medications will come arise from our knowledge of how genetics affects behavior. I'm sure everyone in the room is familiar with how DNA technologies has affected forensics. Knowing that we can use DNA to identify individuals has become an important tool for implicating and exonerating people in crimes, and that this circumstantial evidence of DNA has been very important in adjudicating many different crimes, and I'll get into that in a subsequent lecture. But also, it gives us insight into criminology. And there is a whole aspect of genetics, psychology, that is apart from the criminalistic applications of forensic DNA, but also in terms of its behavioral genetics that relate to certain disorders that might predispose people to violent crimes, but also societally. And lastly, I put on here insurance. All right, right now, they can ask you certain health-related questions. Do you smoke tobacco? Will have an effect on your insurance policy. However, will knowing all the genes that predispose you to certain diseases also render you subject to insurance policy ratings? And there are already laws in legislation to try to preemptively block that idea. But let's face it, if we know that you're going to have a disease and this is going to cost X amount of money, we can bet that there will be strong opposition against those laws eventually. If you could know your genome to clearly decipher your genealogy, would you want to know? Who are you? Where did you come from? People spend an awful lot of time at the Smithsonian looking up old letters and stuff like this. But what if you could know your genome by simply, imagine there was a database that was like Facebook, you know, and rather than being a social network, it was a genomics network. And, you know, you would load up your genome and it would immediately tell you exactly who you were related to all the way back to the dinosaurs. Some people boast of coming over on the Mayflower. Really? Well, perhaps your relatives were dinosaurs. I don't know. You know, does that carry any more prestige? But genomics and anthropology have already been useful for looking at migration patterns. Shown here is the similarity of Y chromosomes and markers related to areas in Eastern and Western Europe. It's been demonstrated that some of the Y chromosome markers in Asia can be traced back probably to a single individual. That would be Genghis Khan. He really got around, apparently. We also can see by genomic comparisons that there is a high level of African diversity which strongly supports the African origin hypothesis for humans, that the origin of humans, that is Homo sapiens, can be traced not only to Africa, but even to certain areas within the African population. So there is more genetic diversity within Africa than the rest of the world owing to this origin, but also identifying the genetics which likely contributed to the migration out of Africa and the subsequent dispersal and migration of humans after that. So we are all African American. This again is that depiction out of Guns, Germs, and Steel showing the presumed migratory pathways of humans, and I think this will continuously be revised as we discover new information. But I think a lot of geneticists, genomicists, and anthropologists agree rather loosely with this map at this time, showing origins out of Africa dating back hundreds of thousands, maybe 1 million years ago or so. Recently, there was an issue in Science that was published on the cave paintings in Spain, showing that perhaps they were not created by Homo sapiens, but rather possibly by Neanderthal, so that admixtures of Neanderthal as they mated with Homo sapiens in Northern Europe certainly were possible. This image shows the distribution of a mitochondrial DNA marker called HVS1 as dispersed between Africa, Asia, and European populations, color-coded there, green for Africa, red for Asia, and blue for Europe. And what it demonstrates is the high concentration of this mitochondrial marker. Mitochondrial markers are maternally inherited, concentrated in the green there for Africa. It's further demonstrating that this marker is associated with an African origin. And then as it disperses throughout Europe and Asia. And then when you look at an individual network comprised of other markers, such as ALU markers, microsatellites, and restriction fragment length polymorphisms combined, you can see that by using a network of individual markers that these different populations can be separated from each other. And if you look closely at this, this is actually a cladogram, which shows the relationship between different groups in Africa and where they separated at the very beginning from those in Europe and later from those in Asia. But the different populations are separated out from each other based upon this, albeit very small, number of markers, let alone what happens when you examine the entire human genome. So genomics gives us a very clear forensic picture of human diversity and human anthropology. And that's actually a backdrop for now how we understand healthcare, because certain peoples will have certain genetic predispositions owing to their background and their populations. So differences in human DNA can be used not only diagnostically to determine disease predispositions, but also for developing individual health care. Let me ask you, if you knew your genome and it revealed that you had a genetic predisposition for a non-communicable deadly disease, say early onset ALS, and that diagnosis predicted your death by age 35, would you really want to know that? Some people would. You might change your lifestyle. You might get out the bucket list early, right? I'm going to jump out of an airplane. I'm going to publish a novel. But really, if you think about it, why shouldn't you have that notion anyway? Using population variation information helps us to understand our anthropological heritage. Also, I would think of it as being an anti-racist tool. Quite frankly, that's my point of view. But knowing that we are all interrelated tells us really that we are all one people, really. And as Dr. Seuss mocked the star-bellied Sneetches, I think we should take this information to heart. So, on the other hand, when we look at it in terms of healthcare, we can understand just as plain as your face that this information can be used for prescribing appropriate drugs and diagnosing disease susceptibilities that, unlike my ALS example, might predict how you might change your lifestyle. If you knew that you were predisposed to type 2 diabetes, would you watch the way you eat? Would you exercise more? How would you change your lifestyle if you knew you had a predisposition for something that you could have control over? This is a diagram showing a very low number of genes that were examined by a commercial group, looking at heritage DNA, showing that we can separate out publicly available DNA by DNA databases, showing separation of African populations, European and Asian populations from Asia, and even Native American populations. And the map and resolution of this kind of technology is increasing all of the time, and having eventually an influence on public health. We know, for example, that the cost of the rising incidence of type 2 diabetes, cardiovascular disease, and obesity will contribute to the ever-increasing burden on our public health system. What should we do about that? Some people have different solutions, whether it's government-controlled healthcare or whether it's left up to the individual. Certainly, this data can't be denied. We are growing older, living longer, and therefore encountering more diseases such as cancer, diabetes, and heart disease. How will we as a culture, as a society, care for these people into the future? These are decisions that we need to make. So from DNA to healthcare, differences in human DNA. How much do we differ? Well, if you look at the number of DNA differences, you would expect that if you isolated DNA from two identical twins, that they would vary by 0 base pairs. These would be monozygotic twins, so when the egg was fertilized and it divided once, copied its DNA and divided once, that those two cells grew into two different individuals having the same DNA. In essence, that's a clone. Two individuals with the same DNA. Now, I will point out at this point, because we will talk about cloning as it relates to animals and to humans later, that if any of you know identical twins, you can tell them apart, both behaviorally and in a lot of cases physically. I have 2 nephews that are identical twins, and it's striking sometimes how different they are. They'd probably laugh if they heard me say this, but nonetheless, identical twins have the same DNA, and the number of DNA differences would be 0. However, unrelated humans, you and you, would differ about 1 in 1,000 base pairs. And those could be single individual base pairs that are different, or large segments of DNA, but the number of DNA differences is about 1 in 1,000 base pairs. The differences between a human and a chimpanzee we now understand is about 1 in 100 base pairs, and we are about 98% the same at the genomic level to a chimpanzee. I said 88% the same as a mouse. And the human versus a piece of broccoli is about, on the number of DNA differences, 2 out of 3 base pairs are different. On the other hand, you might look at it to say that 33% of your DNA is the same as a piece of broccoli. That's interesting, if you flip that around. So this information on population and DNA variants has been used in lots of different analyses. In one shown here, 150 Africans, 120 Europeans, and 700 Asians were analyzed using more than 200 DNA variants measured in each individual. And using these types of analyses, genetic relationships of world populations can be discerned so that you can determine ancestral DNA and compare that to various groups that broke off during this migration, and even now separate out different tribes in Africa, as shown in green here, but certainly distinguishing even separation between the castes in India, owing to the fact that they largely intermarry. The upper caste marries upper caste, and so on. But we can also distinguish between groups of geographically close countries in Northern Europe and markers related to China, Japan, and Southeast Asia. What does this tell us? First of all, that all humans are quite similar to one another at the DNA level. Huh, that's interesting. And that genetic variation is geographically structured. You know, that we can tell the difference between different groups in Northern Europe based on their DNA. And that This is probably owing to the fact that populations that live close to one another tend to be more similar genetically. And this is probably because they marry each other, isn't it? Like, people tend to have babies with people they live near. I guess that's the way it goes. That's becoming less true, but certainly geographic isolation is what caused genetic separation, and so that people in Iceland can be determined as Icelandic. And the entire country of Iceland has been sequenced, more or less. The other fact that this points out is that genetic variation can inform us about the history of a population. What was the admixture? We once thought that Neanderthals did not mate with Homo sapiens. Now we see the inclusion of that genome within our own, as well as other groups like the Denisovans. So this leads us to pharmacogenomics and personalized medicine, the composition of humans. What will the role of pharmacogenomics be on development of drugs? We know that based on a patient's DNA sequence, that one drug that might be good for you might not affect you at all, or could kill him. And it would be good to know that ahead of time, so that drug metabolism even can be studied at this level. So pharmacogenomics is the study of how an individual's genetic inheritance affects the body's response to drugs. And that this type of information then can be used in genetic testing both for personalized medicine ahead of time as well as comparative medicine and used to develop new drugs. So encoded in the DNA sequence of our individual genome is the genetic program to build, maintain, and adapt all of our tissues and functions. And so knowing the complex signaling and regulatory networks will allow us to develop new drugs to treat these disorders appropriately. That was easily said, but look at the signal transduction pathways that might be involved with the complexity of a disease like cancer, or for that matter, HIV. The HIV virus, depicted here, contains a relatively low number of genes and a very short genome that really could be sequenced overnight. For that matter, H1N1 and flu virus can be sequenced very quickly. In the HIV case, given knowing the genome and all of its genes for over 15 years, well, more than that now, we still do not have an adequate vaccine. So the cataloging of humans will allow for genetic testing. Personalized genomes will certainly be a reality. The first genome was over $1 billion to make the first draft. Probably all of you will have access to your own genome within the next decade for under $3,000. I would say probably for under $1,000. It'll probably be covered by your insurance policy and probably controlled by it. No. But this will allow DNA diagnostics. And as someone brought up before this lecture tonight, prenatal testing, if not pre-fertilization testing. Where will this go? What would you want your boy or girl to be? Do you want a boy or a girl? How would we do that? Can we separate out X and Y sperm using flow cytometry and then use that exclusively for doing in vitro fertilization? Do you want a boy or a girl? Then what kinds of characteristics would we add to that? Would that allow then prenatal, if not pre-fertilization gene therapy? If there were a genetic disorder detected in either mom or dad, could we do so-called genetic surgery to replace or add functions? And if that were true, where does that stop? So we could add gene functions, we could delete gene functions. The type of tools are now rapidly becoming available in the box in order to do this to plants, animals, fungi, and bacteria, and yet to be applied to primates or humans. Tissue engineering via stem cells. We've now demonstrated that stem cells can be made available for tissue rejuvenation. What about if we add genes? If you have, for example, a stem cell that's capable of regenerating into a pancreatic cell, why not add an insulin gene that would be properly expressed and cure type 1 diabetes? This is a great idea. Tissue replacements could also be then engineered by making tissues in vitro from genetically modified stem cells. Skin replacement for burn victims made out of their own skin. This is good, no tissue rejection. The biology of aging. Why do we age? Why is that all about? This is certainly biological, and knowing DNA, RNA, and its functions gives us clear insight into the biology of what comes down there. We know, for example, that how DNA is methylated declines with age. Could that be arrested or altered? Could you do that before fertilization and therefore create a human that ages either more slowly or not at all? Actually, I don't know why you'd want to do that, but perhaps future generations will figure that out. And certainly genes and behavior. How much are you a function of your genes? Consider the Tay-Sachs dilemma. This is a genetically inherited disorder in a lipoxygenase that's involved in brain function and development. 2 recessive alleles and the individual will be born to have a very short life and be brain function compromised. So people with Tay-Sachs disease suffer a very short life. If you knew that your baby-to-be was to have Tay-Sachs disease, and this was determined by prenatal testing, what would you do? You could be offered the possibility of starting over. This then brings up the dilemma of selective abortion, a concept that our country, as well as other countries, have difficulty deciding now. What if, on the other hand, through amniocentesis and diagnostics of the DNA, knowing that the DNA in an amniocentesis cell is the same as a fetus, you detect Tay-Sachs disease, a mutation in a lipoxygenase gene which has in fact been cloned. We could take the correct gene and take the DNA, the nucleus from the amniocentesis cell, and do essentially a genetic surgery to either add or replace that defective gene with a functional gene, and use that nucleus to then be implanted into an unfertilized embryo and restart the pregnancy. That means, of course, that we have to terminate the first one and restart this, but on the other hand, it's the same genetic individual with simply 1 gene replaced that now would have the possibility of a normal life. That's a dilemma. That's a dilemma. Our Supreme Court in this country today can't figure out the dilemma that pro-life and pro-choice brings. Now add this. Certainly the technologies will allow this, but then where does this stop? Say like, okay, you have this genetic malfunction of this lipoxygenase gene. What if we simply say, oh, Mrs. So-and-so, you know, we have determined that the projected IQ of your son will be only 120, and these days that just doesn't cut it. But, you know, we have a set of alleles here which could boost that to at least over 140, so he might be at least adequate. You know, so you can see that by some sort of slippery slope argument that we could go from what could be justified as genetic therapy to beyond genetic therapeutics for other purposes, including even those that might be cosmetic or military propelled. So if you knew by genetic testing that your spouse also had the same diagnosis, would you want to even have a prenatal screen? So the ultimate genetic test brings up many questions to consider. Would whole genome sequencing, that is WGS, results be stored as part of medical records for yourself, your offspring, etc.? How will patient autonomy and privacy be protected? Would whole genome sequencing, if done, be done early in life? Maybe at the outset of life? Maybe even before fertilization? Who decides which data can be released until a child is old enough to decide? Who pays for WGS? And if paid directly by patients, under which conditions can they order it directly? Should you be prescribed? If paid by the government, does the government then own the genetic information? How can WGS be made affordable? Is it only for the rich to have access to this? It is also important to guard against the overpromise that this type of diagnosis will bring, especially in the near future until this kind of diagnostics are more clearly worked out, in order to prevent any unethical or illegal use of purported WGS results. I mentioned the mouse genome, published in 2002. Certainly, the mouse genome will provide a great database for looking at drug discovery and disease predisposition. Also, the ability to make transgenic mice, to use cloned fragments of DNA as what I described as being done in plants, can now be readily applied to mice. This means that we can take a synthetic promoter or a promoter from one gene, connect it to a foreign protein, and examine its expression in mice, something that we are unwilling yet to do in humans. We can examine, for example, where is a promoter activated during development in mice by hooking it up to a reporter gene, like green fluorescent protein. When the green fluorescent protein is turned on by that said promoter, it will be expressed developmentally and we can examine its expression. This might be especially important if it's a disease gene, say a cancer promoter. This image, by the way, is digitally created. I need to point that out, but it does exemplify the fact that we can genetically engineer mice. We can turn genes on and off, and we can look at toxicogenomics, another aspect of genomics we might not want to experimentally examine in humans. So personalized genomes are at our doorstep, literally. Okay, are we merely a genetic billiard ball set in motion by fertilization? No, obviously. We are affected by our environment as well. For example, I will show sometimes in class that I can plant two identical pea plants, and if I water one appropriately, it grows, and you have a nice, normal-looking pea plant. If I take the genetically similar, nearly identical seed and plant it in another identical medium and fail to water that one on a regular basis, it might not even look like the same plant. So we say that phenotype, that is what an organism looks like as a consequence of its genetics and its environment, or variation in phenotype equals VG plus VE. That's very simple. So you might have a genetic predisposition, but if you are not then subjected to the environment that would cause that set of genes to be expressed in that manner, you will not end up with that phenotype. Behavior would be an obvious example. Oh, I'm genetically predisposed to violent behavior. There may be certain aspects of your environment which would enhance that behavior. This gets even more wonky, if you will, when you consider complex phenotypes, multifactorial genetics, as well as, not depicted in this slide, multifactorial environments. So VP equals VG plus VE is very simple to understand in terms of a single gene against an environment. But let's consider multifactorial genes, such as quantitative traits. In plants, we would say yield would be one of these. But certainly disease functions like predispositions to cancer, which may involve several different genes for any given organ or cell type, and then by the environment. And we'll talk about cancer biology later, that we talk about field effects, that UVB light on skin, all of your skin cells are subjected to a field effect of UVB. But typically cancer is the result of a clonal event, one single cell accumulating the mutations in order to result in prolonged growth. So having the genes that are involved multifactorially is going to be a very large challenge to the information analysis of genomics, let alone how they impinge on their environment. When do we intervene? Remember, this was a question that's persistent throughout this series of lectures on pharmaceutical biotechnology. Here's the dilemma. How much money would it take to get you to stick a pin in your palm? And let's clarify this a little bit. Let's say it's a clean, sterile needle. There aren't drugs or other organisms involved. And let's call it less than a 20-gauge syringe. So you're not sticking some big, huge thing into your palm, but it's a needle. And let's also say that it's not just a little, you got to jam it in. So there's the premise of this dilemma. I had a graduate student who asked this, and he said $5. He was rather hard up for cash. So, you know, that sets that kind of limit. There are other people I asked who said they wouldn't do it for any amount of money. Why would I do that, you know? And so that kind of sets the parameter. And you can see, well, you can't see what my class said about this, but the results varied from I wouldn't do it to a small amount of money. Let's change this a little bit. How much money would it take to get you to stick that same pin into the palm of a child you don't know? Well, that sounds a little horrible. You're just going to jam this needle into a kid's hand, huh? Well, you could have some sort of predisposition that might allow you to do that more easily than her. I don't know. Some people say, I wouldn't do that for anything. And that's kind of where a lot of people fall when you ask this question. However, I did this to another graduate student of mine, and he said, well, I'd do it for $1 million. And I was kind of like, well, you would? Like, I was pretty surprised because most people would say, I just wouldn't do that, you know? And then he said after I posed the question that, yeah, and then he'd give half of it to the kid, which I thought, that puts a different spin on it. But what if you were to give all the money to the child? And that child then would be benefited by that money and perhaps get out of what might be an otherwise miserable situation. So you can see how this may affect harm versus care dilemmas. Perhaps not the best illustration of that, but on the other hand, you can see that intervention, how we intervene and when we intervene, can be somewhat clouded by situational ethics. So if you knew that you had a genetic predisposition that could possibly be affected negatively by a medication for cancer, would you still take it if it were the best treatment? Kind of like sticking a needle into your own palm. So we already went over what is a genome. Let's look at the different levels of genomic technologies, starting first with the DNA level, then looking at RNA, and then finally protein. We know that the genome location is largely in the nucleus. However, we should recall that there are about 450 genes that are in the mitochondria, and the mitochondrial disorders can be caused by mutations in the mitochondrial genome. So genomics is large-scale collection of biological information on a genome-wide level as it occurs now with DNA, RNA, proteins, as well as their interactions. And knowing genomics will give us a better understanding of diseases, therapeutic targets, predictive medicine, precision therapy, and genealogy. So pharmacogenomics, then again, is that information on human genetic variants that is only just the beginning to be used in drug development. Some of this lecture was contributed by Albert Seymour in pharmacogenomics from Pfizer, and I wanted to recognize him now for his contribution. And he always liked to distinguish between pharmacogenetics, which is the genetic cause of interpatient variability in phenotype, which could give rise to a phenotype owing to clinical symptoms, pharmacokinetic variability, and responses to drugs, as well as genotype, which is the genetic markers that distinguish that variation. So in this diagram, he shows how there may be a difference in the genetic code between responders for a drug and non-responders. And really, that is at the heart of pharmacogenetics as well as pharmacogenomics. A drug that might be good for you might not do anything for you. And that could be determined in the DNA sequence and inheritable. Pharmacogenomics then is looking at genetics as distinguished by SNPs, SNPs, single nucleotide polymorphisms. An A changes to a T, a G changes to a C, and this largely is responsible for variation in humans between you and you. And expression profiling can also occur in changes in regulatory sequences, whether those are transcription factors or promoter elements that cause changes developmentally as well as in response to environment of certain genes. And then how do proteins function in terms of proteomics? So how do we study genomics? DNA sequencing at that level, RNA expression at the RNA level, and differences in protein populations and their interactions. So here are some definitions that relate to this, including expression profiling, which is the study of RNA patterns determining biological functions. Toxicogenomics is recently becoming a larger field, looking at how toxic compounds in our environments will affect each one of us differently. So people have noticed for a long time, for example, that tobacco smoke has a different effect on different people comparing to their exposure level and their genetics. You can think also the same thing of pesticide exposure, flame retardant, BPA, cosmetics. Virtually all of these different pollutants in our environment will have different effects based on our genetics. Pharmacogenomics then altogether looks at phenotype in terms of drug response and disease relating to how we will determine prediction. So the project goals of all of this were to identify human genes, determine the sequence of the individual base pairs, how this information now can be stored. You will think about personalized genomes and their comparison comprising terabytes of information over and over. How's that going to be manipulated? How's that going to be stored and accessed? I think Moore's law will predict that we'll get better and better and better at this. And develop faster and more efficient sequencing methods so it can be done cheaper and faster. And develop tools for data analysis. And then eventually, how will we think about ethical, legal, and social issues, or ELSI, that may arise from this project? Okay, out of our 23 chromosomes and 3 billion base pairs, is this the blueprint for DNA? I think we've already decided, no, not exactly, as it interacts with our environment. But the decision really now is not whether to sequence the whole thing or just functions of it, like coding sequences, because now sequencing the entire genome is rather quick, owing to technologies that are now present, like Illumina, 454, and related sequencing technologies. But this will continue to improve. Facebook, you know, is already obsolete. I'm saying that because in a moment's time it will be. Similarly, these sequencing technologies. I could say Illumina and 454 are the thing today, but really being able to sequence a genome is becoming continuously improved. Recently, nanopore technologies have just been released owing for very fast and accurate DNA sequencing. So originally, making physical maps and cloning these in terms of using bacterial artificial chromosome or BAC libraries allowed people to shear a human chromosome into individual fragments and then sequence them and use computer-driven algorithms in order to stitch them back together. So you can see in this diagram that taking an entire chromosome, shattering it into larger bits, and then including those into bacterial artificial chromosomes, and then sequencing the ends using paired end reads, we can overlap these and stitch these back together. This is wonderful because remember the old movie Jurassic Park, right? The mosquito lands on the dinosaur, takes up some dino DNA, and it's amplified. Well, when that movie was made, there was no algorithm in order to stitch dino DNA back together and reconfigure its structure. Because if dino DNA were amplified and subjected to sunlight at all, it shattered into all these little bits. And any geneticist that would criticize that movie in the old days say, oh, that's impossible, they can't do that. You know, they can't stitch this all back together. Well, lo and behold, this is not far-fetched at all. And those same algorithms allow us to do this out of DNA that's been in a Neanderthal femur for 18,000 years. We will be able to sequence the DNA of organisms long gone. And, you know, don't you think that we would be able to synthesize this? Surely. Will we be able to bring back dinosaurs? I don't know. I think I'm amazed that we could sequence Neanderthal DNA. The Human Genome Project was conducted by 5 large consortiums organized by Francis Collins, the DOE, Baylor, Sanger Center, the Whitehead Institute, and Washington University Sequencing Center. And now there are other large genome centers around the world. The private company, Celera, led by J. Craig Venter, really put the feet to the fire of the public group, and they both accomplished it at roughly the same time. And now we have the technology that's been accelerating since. This pie chart shows the distribution of the different proteins that have been mapped out of the Human Genome Project due to their different functions. You can see on the list there are structural proteins, there are receptors, isomerases, hydrolases, phosphatases, transcription factors, etc., and the percentage of still unclassified genes. This is constitutes the coding region in the human genome. While the human genome was sequenced in its rough draft form in 2001, it was completed with a 1 in 10,000 error rate in what was called a finished draft in 2003. Where did our information come from originally? How did it change? Who are we? How do we interpret the information? When that April with her sure sota, the drought of March was pierced to the root, and it bathed every vein in sweet liquor, of which Vertur engendered is the floor. And Zephyrus eke with his sweet breath, inspired hath in every holt and heath, the tender Acropus and the young Gasona, hath and the ram and half coor Zerona. And small fowls speak in melodia, and sleeping old and neat with open ear. So pricketh them in nature and here courages, the long and folk to goon on pilgrimages, and especially from every Girizenda to room 100 and CBLS de Venda. What is that? Does anybody know? What language is that? Well, I get guesses in my class that that's German or some other language like that. Actually, that's from Canterbury Tales. So that would be Old or Middle English, and you can see in this translation how it compares to us. So where did our language come from? Was it always English? You can tell by different dialects how language is today still shifting. You can compare a dialect from southern United States to that of Minnesota and see striking differences. Or in Rhode Island, where there is, or places on Long Island, a confusion of A's and R's. One student came up to me, for example, once and said, "Dr. Kausch, it'll never work. It's been under the water forever." Under the water forever? Really? Or have one student at Brown asked another student if she had had her ESP-est. And the student said, ESP-est? Yeah, ESP-est, you know? Anyway. You can see how language is mobile. Well, so is DNA information. And it changes over time, and it changes relative to our environment, and it will be geographically subjected to the same selection pressures. Interpreting the genome. So you have your A's, T's, G's, and C's. That's cool. A's, T's, G's, and C's. We know 3 billion of them. What does that mean? You know, that's like if you looked at this and it's just a preponderance of an alphabet. Until you can actually apply the appropriate filter to understand the meaning of the language that is inside this, where once upon a midnight dreary while I pondered weak and weary over many a quaint and curious volume of forgotten lore and on, so that out of this continuous information falls out meaning. So out of the A's, T's, G's, and C's, we can now distinguish coding regions by their unique footprint. It turns out also that some of the genomics tools are now being applied linguistically, which is, I think, another interesting utility. And in fact, words and language have a direct parallel to the information in biology. From DNA to humans. Traits, as described as early as Mendel in the 1800s, that encode P's and their characteristics are inheritable and assigned genetic function. The same is true for human genetics. Sequence polymorphisms can be used to mark different genetic traits that are observed in a phenotype. So genotyping technology then is a molecular tool to quantitate those polymorphisms. So the types of single nucleotide polymorphisms that may occur in a human are these SNPs, where one base pair may change to another during errors in replication and be inherited, and mark or cause variation in gene sequences. Indels, or insertion deletions, can occur at the base pair level or whole sequence or whole chromosome arm level. And SNPs are mapped according to individuals at this point. You can take a single gene locus, like epithelial growth factor in humans, located on chromosome 7, and map for the SNPs that are present in different individuals and relate those to different disease phenotypes. So the effect of SNPs compared to what is shown here at the normal sequence you can get a single base pair change shown here, which will be silent, where that amino acid TCA codes for the amino acid serine. The substitution of another C because of the redundant code, TCC, also codes for serine and is therefore a silent SNP, but still a base pair change. On the other hand, you could have a conservative missense mutation where that T is transferred into an A, and now you have ACA, which codes for not serine anymore, but threonine, which may cause a different function in that protein, but maybe not likely because serine and threonine are similar. You could have a non-conservative missense mutation where that C change there causes that to become a proline. Or a nonsense mutation where a substitution of that C for an A codes for a stop codon, and then therefore a truncated protein, and therefore a protein that probably doesn't work very well, if at all. Or a frame shift mutation, where the insertion or a substitution of a base pair there causes a misread and hence an entire change in amino acid sequence downstream. So SNPs can have small effects, no effects, or large effects. And we can manipulate the genome accordingly. And we can score for these SNPs by large-scale human genome sequencing, and we'll be able to apply that information accordingly and help us to find disease genes. So comparative genomics then will give us insight into individual gene function as well as into our relationship and function of in other organisms. For example, mice and humans, indeed most or all mammals, including dogs, cats, rabbits, monkeys, and apes, all of which have been sequenced, have roughly the same number of base pairs in their genomes, roughly about 3 billion base pairs or so. The most significant differences then between mice and humans are how those genes are expressed, not in the number of genes per se, but in the structure and activities which regulate the gene expression. So gene for gene, we're very similar. How can we use the genome sequence across species to find new genes? Well, we might want to look at which genes are most highly conserved. For example, here, if you were to take the entire human genome and look at a comparison between the human and the mountain gorilla genome, you would see overlapping homology in certain regions, and you could then identify similar homology in mice, and that this homology may show you a conserved region which may be important owing to its conservation alone. This isn't always true. There are a lot of examples which might point out otherwise, but certainly conservation through gene family homology is important and might identify new genes involved in certain important functions so that mutations in those genes could be identified as important for disease or drug target identification. So comparative genomics then helps us discern how organisms diverged in speciation and became new species. And what we're doing right now in our switchgrass research also is a clear example of that kind of an analysis. But what do the genes do? Is in my example of the Edgar Allan Poe poem, functional genomics, and discerning what the individual genes do is part of the hard work now of genomics. What is assigning function and dysfunction to that genetic sequence is what the hard work is of functional genomics, not only in terms of its RNA, but its protein. So imagine that you know all the information in a phone book for New York City. That tells you the name and address of the people who live in New York City. but it doesn't tell you what they do for work, what they do when they leave their house, their address posted in that phone book or whatever. It doesn't tell you their relationships on a day-to-day basis, or how they interact, or maybe if they do or do not have children, and so on. So knowing the function in the network of all of those different people in New York City is by far more complicated than simply knowing the information which attaches them to a certain address or their last name. So you can see how complicated then by that analogy this gets. How do we study genomics? Well, I've already told you about DNA sequencing. And if we look at RNA expression, we see that there may be 200,000 different exons that can be arranged or rearranged to code for maybe over 80,000 different proteins, all which may be exerting collectively different traits. So looking at the role of RNA as it relates to disease traits is certainly an important part of this analysis. In other words, there might be certain genes that are on in disease states that are not on in disease-free states. So knowing the genetic profile of gene expression as a tool becomes important, and looking at the RNA population is a part of that. So we can arrange to look at RNA populations by taking sequences of DNA and then hybridizing RNA that's expressed from certain cell types and illustrating then which genes are expressed in that particular population. This technology is called microarrays or DNA chips. So these so-called biochips or microarrays are used for expression profiling, which is the study of RNA patterns, which could help elucidate various biological phenomena and gene expression appropriately. So microarray technology then is reshaping how we think, or how certainly we analyze gene expression, not only developmentally, because some genes that are on early in development are off later, some that are related to disease traits during development may not affect an adult. We know lots of different diseases that are characterized as childhood diseases, so-called, and that a person grows out of these because of the expression pattern related to that. Or it may work the other way around as well. So in gene chips, each spot shown on an array like this contains a different gene sequence. So a gene then, in terms of its complementary DNA, its cDNA, can be posited as a single strand of DNA on this chip and then hybridized to its RNA population. So wherever an RNA then, which it might be labeled fluorescently, is complementary to a gene, we would say that gene is on in that cell type. So how does this actually work? You can see in this cartoon that if we prepare a cDNA probe and make this microarray, we can use RT-PCR and label up RNA from normal and diseased tissue, so that we could label these with different fluorescent dyes, shown here on the left in green and on the right in red. And we combine these in equal amounts and hybridize them, these probes, to the microarray and scan, so that the normal tissue is green, the tumor tissue here, the diseased tissue, will come out in red. And demonstrated here in this figure, actually from a published article on breast tumor analysis, showing that in the disease-free tissue, and you don't need to analyze this spot for spot, but just overall you can see a marked difference between the green spots on the upper part of this array and a metastasizing tissue shown down here, which has a large concentration of red spots owing to the different population of RNAs present in those cells. So you can see that this type of analysis can tell us a lot about disease characteristics, but even right down to what differentiates one type of tumor from another type of tumor. And then how will that be treatable? So these so-called biochips then are also called microarrays, and I just wanted to clarify that when you look at this question. So they're not genetically modified potatoes. I had mentioned junk DNA, that there is DNA that does not code for proteins, but it was discovered even before 2002, but commemorated in this cover article of Science, new roles for small RNAs. This was discovered through a sequence of different investigators. I would say started by Rich Jorgensen, a plant biologist working at the University of California, who looked at the silencing of certain genes in transgenic petunias. This went on to lead some researchers working on the roundworm C. elegans to actually feed roundworms RNAs that were associated with development of certain cells, noticing that by feeding them specific genes, they could stop gene expression, owing to the fact that they could now use RNA that would block expression of RNA, and thereby the discovery of so-called RNA silencing or RNA interference. So RNAs would hybridize with our existing messenger RNA, preventing their translation and their expression. So what we see is that there are all these small RNAs then that are regulating gene expression. So these things that were previously thought to be junk DNA actually are very involved with regulating which genes are on and being expressed and which genes are off. You can make a mature messenger RNA which is not expressed then into its protein because of the regulation of these types of RNAs. So RNAi then was subsequently discovered, or these short interfering RNAs, also called siRNAs, that are pieces of double-stranded RNA that may be only 21 to 23 base pairs long. And the biology then is described in this diagram on the left about how RNA actually works. And the reason I bring it up in the context of this class is that you could see that the application of being able to control gene expression using RNAis could have therapeutic applications. You want to shut off a gene? Is it possible that an RNAi might be developed as a drug? How do you deliver that might be another question, but certainly the application of being able to control gene expression at this level will find important applications, and the people involved in this received a Nobel Prize for its elucidation. So pharmaceutical biotechnology now has gone beyond small molecule drugs, beyond recombinant DNA transgenes to make antibodies and monoclonal antibody drugs like Erbitex or Herceptin and vaccines, but now into the development of individual molecules which can control gene expression. Now on to proteins. We can realize the population of proteins by gel electrophoresis in 2 dimensions. Single dimension protein gel electrophoresis will show bands of DNA as they reach their isoelectric points within a gel. However, if you run them in 2 dimensions, you get arrays of spots shown in this slide here, which correspond to individual small protein changes that occur either developmentally or within disease traits or disease predispositions. So looking at which proteins actually are present is similar to which RNAs are present, but these proteins now we can study how they interact with each other in disease functions as well. So proteomics then is sort of looking at the ecology of proteins. So we can look at which proteins are expressed when, but also deduce signal transduction pathways. One protein turning on as a signal a cascade of gene expression that might be involved developmentally or in disease functions. All of this information, imagine, I said terabytes of information associated just with DNA comparisons. Now you have RNA comparisons, protein comparisons, signal transduction pathway comparisons. This gets to be an awful lot of information, so Eric Lander's prediction becomes true. Biology is rapidly becoming an information-based science. The career of bioinformatics did not exist a decade ago. I don't think the word existed 15 years ago. Now this will become a large function of biological sciences as we move into the future. Thanks a lot.